Hi folks, this is Mr. Wendland, and today we're going to be looking at the physiology of the echinoderms. As you can see from this slide, there's some sea stars on it, and those are echinoderms. So we're looking at that particular group today. The echinoderms are very common. We see them quite a bit in tide pools and sandy beaches and down on coral reefs. So pretty common animal. Let's take a little dive deeper into them and see what we can find out about their physiology. So to begin with, sea stars are pretty advanced creatures. Developmentally, they're not that much different from a human. They have a lot of the same characteristics that we have as we're developing. However, even though they are very advanced, they don't have circulatory systems. They don't have blood. They don't have a very complex respiratory system. They have very simple gills. That's about it. They don't have excretory systems. They don't have kidneys or anything like that to regulate waste. They do not have a very robust nervous system. The nerves are there, but there's no brain, there's no head or anything like that. So very primitive animal. And finally, they can reproduce asexually. That right there is a primitive characteristic. Most advanced animals reproduce only sexually. However, the sea stars can reproduce asexually and regenerate. If a sea star loses an arm, it can grow it back. If a small sea star gets cut in half, it can even happen where you have two identical sea stars. So these are things that have very primitive characteristics, yet developmentally are quite advanced. So let's look at the three characteristics of them. The first characteristic that all echinoderms have is the adults exhibit pentaradial symmetry. You should get familiar with that term. Pentaradial means 5 radial, so we can cut them in half exactly 5 ways. 1, 2, 3, 4, and then this way 5. It's a lot different than radial symmetry, where we could cut them in half in infinite numbers of ways. So these are kind of similar to that kind of symmetry. They do have oral and aboral sides as opposed to right and left, top and bottom, things like that. But these still are classified with the bilateral animals. And why is that? Well, it's because the larva is bilateral, and we can cut it. We can see if we put a line through it, we can cut it in half exactly one way. So pentaradial symmetry is the number one characteristic of these echinoderms. Now the number two characteristic is something we can't really see. So if we look at the external echinoderm here, this is just a sea star, we can't see this characteristic. It's actually in its development patterns. All we see when we look at one of these, we see an external mitochondrial, which we'll talk about in a while. We see a central disc, some arms. You won't see the anus. It's very small. It's just a small little opening. But they're very simple animals. And here is the larval form. So what is this characteristic that you can't see with the echinoderms, but is definitely there? Well, that characteristic is deuterostome development. Now, deuterostome is a word that maybe you haven't seen before. It means the anus forms first. And that sounds kind of silly, really. The more advanced animals have an anus form first, and the more primitive animals have a mouth form first. That, however, isn't kind of the big characteristic of this whole development. That's just how development takes place. And it kind of makes sense because I guess primitive animals, some of them don't have a one-way digestive tract. They only have a mouth, things like cnidarians and jellies and anemones. But as I said, that's not really the big issue or big thing about the deuterostomes. What the major characteristic of these is how they develop. Now everything starts off as a single cell and then starts developing from there. It goes from 1 cell to 2 cells to 4 cells to 8 cells and so on. When a protostome is developing, we'll look at the protostome first. It goes from 1 cell to 2 cells to 4 cells. And then if you look at the 8-cell stage here, things are kind of not lined up. It's kind of like you took a Rubik's Cube and you took the top of it and you twisted it slightly. So if that was the case, you couldn't really turn the Rubik's Cube. Things are not aligned. Now compare that to the deuterostome development over here. So if we look at the deuterostomes, at that 8-cell stage, everything lines up, and this is called radial cleavage. Everything is nice and aligned. Now the big thing is, when these things are developing, if you're a protostome and you're primitive and one of those little blastomeres, one of those little initial cells falls off, development is arrested, which means it doesn't take place. The thing aborts right there and you don't get an animal out of that. However, with the more advanced characteristic, the deuterostome characteristic, if one of those blastomeres falls off, it actually can form two normal larvae, and it has to do with how these things can just cut in half this way and perfectly fall apart, and all those cells are touching so they can help repair and reproduce each other. So you get something with advanced animals that you cannot get with primitive animals, and you've probably guessed it, and that's twins. Primitive animals can't make twins. Something falls off, the thing just aborts. If you have an advanced animal, however, you can get two normal larvae even if something goes wrong, and this is likely why the advanced animals don't have to produce as many eggs as the primitive animals, because they're a lot more viable, the embryos. So those are the first two characteristics. One is pentaradial symmetry, and the second one is deuterostome development. Now the third one here is that they have an endoskeleton. They actually have an internal skeleton of calcium, just like we do. We call them ossicles. Ossification is the term for the formation of bone, and these dermal ossicles, you can see they sit right here, in the dermis. Let me go back to that slide. Right here in the dermis, so right in the skin here. They're kind of embedded in there. And if you've ever seen a dried out sea star, it's actually these ossicles on top. The outer layer of skin is just missing. Now underneath these, there's some muscle and things like that. So they are somewhat flexible, but they're the first animal where we actually see this endoskeleton, and that's quite the advanced characteristic. We have the same type of skeleton. So just to recap, the three characteristics of echinoderms are this endoskeleton, deuterostome development, and that pentaradial symmetry. So let's kind of switch gears a little bit here, and we'll go inside. Now inside an echinoderm, it's very interesting. They have something called the water vascular system, and that's something that is very unique to this group. Basically, there's a bunch of tubes and canals in there, and what they do is they're all connected, and they use water pressure to move things around. The water vascular system is super important. They use it for locomotion, they use it for feeding, they use it for gas exchange, and it's really kind of an ingenious system. If you think about slowing a car down, it takes a lot of power. That's why we use hydraulics. We use brake fluid. That fluid pumps into those tubes, and what it does is it squeezes your brake calipers to slow your car down. If they were just made of air, it would take a lot of power and a lot of compressed air to do so. But with liquid, it's much more efficient. So these are very, very efficient animals. And if we take a look at this water vascular system, it runs throughout the body. Now on the outside of a sea star, in one of our initial slides, you saw kind of a round area called the madreporite. That's the mother pore. That's where the whole system kind of starts. Now it's on the aboral surface, so opposite the mouth. Water comes in this, goes into something called the stone canal. Now the stone canal doesn't have rocks in it. It's named after a guy named Stone that discovered it. And then water goes into this central disc and fills up the ring canal, and this is where most of the water is stored. There's also these little pulling vesicles right here, and what they do is they can store extra water. Think of like a water balloon. If you were to have a little extra water to pump in there, you could squeeze that water balloon and it would turn left or right or become more rigid. Now, all this stored water can be moved into these radial canals that radiate from the center whenever the sea star wants to move one of its arms, and it ends up in these little ampullae, which are these little sacs above the podia, which are the tube feet. So we'll take a little closer look at that. But the tube feet usually line these grooves on the bottom. They're called the ambulacral grooves. And this is where the sea star stores all its tube feet, and this is where it gets all its motion and movement from. So you should know the order that these things, the water passes through these things. And to make it easier, I've given you a mnemonic. It says, man should rock and roll and party. And that's how you remember it. Water comes in through the madreporite, goes into the stone canal, fills the ring canal, down the arms through the radial canals, into the little ampullae, and finally into the podia, which are the tube feet. And that's where ultimately the sea star movement comes from. Now, if you took a look at one of these ampullae and tube feet, you would see something that looks kind of like this. So this water coming down the radial canal came from the ring canal, and it fills up this ampulla. There's a small lateral canal there, and it fills up the ampulla. The ampulla is kind of like a sac, and there's muscles in it. So this is full of water. When those muscles squeeze that ampulla, it can cause this foot to go right or left, and it can individually control each one of these. So rather of an ingenious system, it's very powerful. It just uses the surrounding water to fill it up, and then the muscles squeeze that water and force it into areas, and it has a lot of pressure. Think of squeezing a water balloon. If you have a very long water balloon and you were to squeeze it, the thing would bend to the right, to the left. Or think about a hose. If you turn it on and let go of it, man, that's a lot of pressure. Sometimes those hoses whip back and forth. And if you have a lot of pressure, it can cause a lot of movement. And that's exactly what the sea star uses to its advantage, or the sea urchin, or anything else in this family for that matter. Now, sea stars are what we mainly think of when we think of echinoderms. You can see the tube feet on this one. Sometimes they have big tube feet with suckers. They almost all have suckers, and sometimes they have a small amount of tube feet with all this skin in between, like bat stars. Sometimes they just have a boatload of tube feet all over the place. Even sea urchins have tube feet in between their spines. So they all use this water vascular system to move around, and for locomotion and for gas exchange and things like that. All right, so that is the physiology of the echinoderms. Next time we'll go through their classification and we'll see how they feed. It really depends on the class how they do that. So until next time, have a great day, and we'll talk soon.